Hi viewers, today I will discuss important topic that is called PCR. PCR is an abbreviation of polymerase chain reaction. It is called a chain reaction because the products of this process are used again and again. So first of all we will discuss here what is PCR. Method. PCR is a method used to rapidly make millions to billions of copies of a specific gene or small part of DNA in vitro. In vitro means in laboratory. PCR is a laboratory method in which we can generate millions and billions of copies of gene or small part of DNA. Discovery. PCR was developed in 1983 by Carey B. Mullis. Carey B. Mullis was an American biochemist who won the Nobel Prize for Chemistry in 1993 for his invention. PCR has some advantage over conventional method. Before the development of PCR, the methods used to amplify or generate copies of recombinant DNA fragments were time consuming and labor intensive. In contrast, a machine designed to carry out PCR reactions and producing billions of copies of DNA fragments in only a few hours. Materials requirement in PCR process. There are the materials that are required in PCR process. Number one, gene or specific part of DNA that have to amplify. Number two, primers. Number three, DNA polymerase enzyme. And number four, free nucleotides. And number five, cofactor. And number six, buffer solution. Polymerase enzymes. Initially, PCR was performed with DNA polymerase 1 of E. coli as the replicase. But this enzyme is heat inactivated. During the denaturation step, fresh enzyme has to be added at step 3 of each cycle. Taq polymerase. This enzyme was discovered in thermophilic bacterium that is called Thermus aquaticus. It remains active during the heat denaturation steps and does not have to be added after each cycle of denaturation. It can tolerate temperature up to 120 degrees centigrade and found in hot springs. Taq polymerase limitation. It does not contain a built-in 3 to 5 proofreading activity and consequently it produces a higher than normal frequency of replication errors. If an incorrect nucleotide is incorporated during an early PCR cycle, it will amplify it just like any other nucleotide in the DNA sequence. It amplifies long tracts of DNA greater than few thousand nucleotide pairs inefficiently. High fidelity in PCR. When high fidelity is required, PCR is performed using heat-stable polymerases. PFU from Pyrococcus furiosus, TLI from Thermococcus litoralis. Both contain 3 to 5 proofreading activity. TFL polymerase from Thermus flavus is used in case of long segment of DNA up to 35 kb in place of Taq polymerase. Steps. 3 fundamental steps are involved in polymerase chain reaction. Number 1, denaturation. Number 2, annealing. And number 3, extension. Denaturation. The genomic DNA containing the sequence to be amplified is denatured by heating to 92 to 95 degrees centigrade. Annealing. The denatured DNA is annealed to an excess of the synthetic oligonucleotide primers by incubating them together at 50 to 60 degrees centigrade. The ideal annealing temperature depends upon the base composition of primers. Extension. DNA polymerase is used to replicate the DNA segment between the sites complementary to the oligonucleotides primers. The primers provide the free 3' hydroxyl group required for covalent extension. The denatured genomic DNA provides the required template function. Polymerization is usually carried out at 70 to 72 degrees centigrade for 1.5 minutes. Role of magnesium chloride. Essential cofactor that enhances the activity of Taq DNA polymerase, which in turn increases the amplification rate of DNA. Magnesium chloride increases the amplification rate of DNA. However, higher concentration of magnesium chloride can result in decreased specificity. Here is a process that consists of whole PCR mechanism. The parent DNA is separated in first step by denaturing at 95 degrees centigrade. The two strands of the DNA are separated from each other. In step 2, annealing at 55 degrees centigrade, Taq polymerase, free nucleotides, DNA primers all are mixed in the process. Step number 2, that is called annealing. Here is the primer at, and this primer is at the 3 end of the DNA. In step 3, that occur at 72 degrees centigrade. The whole process occurred in the 3 to 5 direction, 3' to 5' direction, and the 4 DNA strands are formed from the 2 strands, and the process is repeated again and again, and this repeat cycle 20 to 40 times, and it creates millions and billions of copies in just a few hours. The whole process describes the PCR mechanism. Application of PCR. Here are the applications that are used in basic research and in applied research. In basic research, PCR is used in mutation screening, drug discovery, classification of organisms, genotyping, molecular archaeology, molecular epidemiology, molecular ecology, bioinformatics, genomic cloning, site-directed mutagenesis, and gene expression studies. In applied research, it is used in genetic matching, detection of pathogens, prenatal diagnosis, DNA fingerprinting, and gene therapy. Thank you very much. Please subscribe my channel for further videos. Thank you all of you.